Living creatures move about in a variety of ways. On land, animals use their legs. In the water, fish flex their bodies, tails, and fins. In the air, birds and insects use their wings. No creature visible to the naked eye, however, uses a rotating mechanism for movement. It was under a microscope that such life forms were found. Many bacteria were using rotating helical flagella to swim about. The discovery of the rotary motor mechanism in bacterial flagellum, reported by American researchers in 1974, sent shockwaves to the global biologist community. Such biological mechanisms are found on the scale of nanometers, or 1/1,000,000th of a millimeter. Today's most advanced fine processing technology is still trying to advance to sub-micrometer fabrication. However, finely designed nanomachines have already been perfected by nature. The setup of the flagellar motor, which looked simple at first, was rather complex. Scientific and technological progress was needed before its mysteries could be unraveled. A Nobel Prize was awarded to Paul Boyer and John Walker in 1997 for the discovery of the rotary catalytic mechanism of ATP synthase. However, the rotation mechanism of flagellum has still not been clarified even after nearly 30 years. The Japanese government regards nanotechnology as a field of priority in its basic plan of science and technology begun in 2001. Well ahead of this plan, the Japan Science and Technology Corporation, JST, launched a research project on the bacterial flagellum to look into the mechanism and future application of biomolecular nanomachines. In 1997, JST began the Namba Protonic Nanomachine project as part of its Erato program. The project is under the direction of Dr. Keiichi Namba, then Research Director of the Advanced Technology Laboratories of Matsushita Electric, and now Professor of Osaka University. The project has been studying the structure and function of the flagellum as a huge molecular system on the basis of the organization and movement of the individual atoms that build it. This requires extremely precise and laborious work. Salmonella, the main object of study, has several flagella. With a motor at their base, they are only 20 nanometers in diameter, but are over 10,000 nanometers in length. The flagellar motor is similar to man-made motors. The flagellum consists of rotor and strator units in the cell membrane, switching unit, bushing, universal joint, helical screw propeller, and so on. It rotates at a speed of up to 20,000 rotations per minute, which is faster than the speed of Formula One race car engines. The motor is driven by protons flowing into the cell body. Its energy consumption is 10 to the minus 16th watt, and its efficiency above 80%.
Flagella are biological machines, with each flagellum made up of about 30 different proteins, numbering from several to tens of thousands. They all work together as one unit, In order to understand the overall mechanism of the movement of the unit, which consists of billions of atoms, it is necessary to study the three-dimensional arrangement of all of the atoms that make up this biological apparatus. You need to check the dynamics of individual atoms. This is a major challenge, and not many people believe that this can really be done. However, without understanding this, without observing what is going on, there is no way to understand the dynamics or structure of the protein nanomachines.
The project consists of 3 research groups and a project office. The nanoassembly group analyzes the molecular structure of flagellar proteins by X-ray crystallography and fiber diffraction. The nano-switching group studies, by electron cryomicroscopy and image analysis, the structure of large protein complexes that are impossible to crystallize. The nanomechanics group measures the rotation of and proton flow through individual flagellar motors. Close collaboration between the groups is essential to unveil the mechanisms of self-assembly, torque generation, and switching of the molecular motor and bio-nanomachines. How can the helical structure of the flagellar filament be formed in the assembly of a single protein, flagellum? How does it change its helical hand from left to right at the moment of cell tumbling? The project team began research in order to clarify such unidentified mechanisms. The flagellar filament is a tubular bundle of 11 protofilaments, each of which is an axial array of flagellum subunits. The inner double cylinder and outer protrusion are made by each flagellum domain. In 1970, Sho Asakura proposed a 2-state protofilament model in which the 2 helical states have slightly different repeat distances. The Namba group, which began studying the flagellar structure in 1986 and continued research in the Erato Hotani project and later at the International Institute for Advanced Research Matsushita Electric, clarified the basic structure of the flagellar filament. In 1998, it accurately determined by X-ray fiber diffraction the 2 repeat distances of the 2 protofilament structures as 52.7 angstroms and 51.9 angstroms. Surprisingly, a very slight difference, only 0.8 angstrom, was responsible for the helix formation of flagellar filaments. Different combinations of 2 protofilament states with different repeats result in various helical structures. When the motor reverses instantly, shear stress is induced in the flagellum, which causes a small sliding shift between neighboring protofilaments. Then, several protofilaments switch into a state of shorter repeat, and the helix changes its hand from left to right. The bundle of flagellar filaments falls apart, and the cell tumbles by the force of unbalance. To understand where the accurate length switch mechanism was embedded, the molecular structure had to be determined at an atomic level. The nanoassembly group used X-ray crystallography for this purpose. To determine the precise molecular structures of flagellum and many other flagellar proteins, they were cloned, produced on a massive scale, purified, and subjected to crystallization screening. However, since proteins that form the flagellar axial structure have a strong tendency to form fiber structures, all attempts to grow crystals in which the molecules are arranged in three-dimensional lattices failed. They only resulted in fibrous aggregates. So, for each protein, the group determined the regions of the amino acid chain that stabilize the fiber structure, produced fragments lacking these regions on a massive scale by genetic technology, and collected and purified them for crystallization. In the case of flagellum, which consists of 494 residues, a fragment named F41 with both terminal regions of about 100 residues in total truncated gave a good quality crystal for the first time. Efforts spanning 10 years were necessary to successfully grow this crystal. Today, it is possible to grow crystals for the majority of flagellar proteins through the same procedure. Even with F41, achieving crystallization was not a straightforward process because it had a tendency to form many small crystallites. The group members had to repeat a tremendous amount of trial and error before a very specific condition was found. The reason for this difficulty was not understood until the structure was clarified. The group faced other difficulties as well. Since the crystals of F41 were only several micrometers thick, X-ray diffraction measurement was extremely difficult. Conventional X-ray apparatus were of no use. Fortunately, a high-brilliance, fine-focus X-ray source that runs stably had been developed at the beginning of the project term in anticipation of such difficulties. Improvements in the crystal freezing and annealing techniques allowed high-resolution data to be collected. Nevertheless, even the selection of crystals that give high-resolution diffraction data was extremely hard work.
With the crystals we had been dealing with up to then, 15 to 30 minutes of exposure time was sufficient. But in the case of this crystal, 6 hours was needed to see the diffraction spots. That meant only a maximum of 4 images could be obtained in 24 hours. But we needed at least 180 images in order to collect one set of data. With the huge amount of time necessary to get just a set of data, it would have been impossible to complete this work within the time frame of the project.
In an effort to achieve a breakthrough, the group collected diffraction data by visiting ESRF, a synchrotron facility in Grenoble, France. However, Spring 8 beamlines, which produces the world's most powerful X-ray beam, opportunely began operation in Harima, Hyogo at that time. A set of data of sufficient quality was successfully obtained by using this facility. A three-dimensional atomic model of F41 was finally built at 2 angstrom resolution from the diffraction data obtained by using a brilliant X-ray from the trichrometer optics installed in a RICAM beamline at Spring 8. This figure shows the structure of F41 in ribbon representation. It consists of 3 domains: D1, D2, and D3. Domain D1 is made of 3 long alpha helices and 1 beta hairpin structure. Domains D2 and D3 are mainly beta structures. The repeating period along the A-axis of the crystal was found to be 51.9 angstrom, which is exactly the same as the shorter repeat of the flagellar protofilament. The A-axis array of F41 in the crystal was superimposed on a density map of the flagellar filament that had been deduced by electron cryomicroscopy and image analysis. The fit was perfect. The protofilament structure was maintained even in the crystal. The crystallization of F41 had been difficult because the ability of flagellum to form fibrous structure had remained in part. Because nucleation took place rapidly in the solution, it had resulted in many small crystallites, preventing the growth of large crystals. The helical shape of the filament switches by switching the protofilament structure between 2 states. But it was not possible to identify the region that works as this lengthwise mechanical switch simply from the structure of F41. What was found, however, was the protofilament structure of the shorter repeat. So, the group conducted a computer simulation of the protofilament extension to find the structure responsible for the high-precision mechanical switch. Three axially aligned F41 molecules were used as a model protofilament. The top molecule was fixed, and the one at the bottom was pulled down by 0.1 angstrom at every step. The deformation of the middle molecule was examined through energy minimization. In the early stages, the molecule in the middle was simply gradually vertically elongated. But then, an abrupt conformational jump took place at the beta hairpin structure of domain D1. The downward shift of the beta hairpin appears to work as the mechanical switch to change the repeat distance of the protofilaments. The change in the flagellar helix is thus induced by such subtle structural changes in the molecular structure of flagellum. These results were published in Nature and featured on its cover. It greatly stimulated the research of the mechanical properties of proteins. One of the remarkable characteristics of a bio-nanomachine is its ability of self-assembly. The bacterial flagellum assembly process starts from an MS ring formation in the cytoplasmic membrane. Then, a switch complex called the C-ring is assembled on its cytoplasmic side, followed by integration of the protein export apparatus inside the ring. The export apparatus sends out flagellar proteins from the cell body to the distal end of the flagellum to grow the structure. Next, the hook, working as a universal joint, extends to the outside of the cell. Then, the two junction proteins HAP1 and HAP3 are attached, followed by the binding of the cap protein HAP2 to form a capping structure under which the assembly of flagellum molecules begins to grow the flagellar filament. flagellum molecules are inserted just below the cap successively, and the flagellar filament continues to grow. All of the flagellar axioproteins produced in the cell body are sent into the central channel of the flagellum and transported to and polymerized at its growing end. to 30,000 flagellum molecules polymerized to build a 10 to 15 micrometer long filament. However, it was not possible to crystallize the flagellar filament or cap filament complex for use in X-ray structural analysis. Therefore, electron cryomicroscopy and image analysis were used to analyze the molecular structure. The cap filament complex was embedded in glassy ice by quickly freezing in liquid ethane a thin film of sample solution on a grid. It was examined by electron cryomicroscope at liquid helium temperature around minus 270 degrees Celsius. High-quality images with relatively little distortion were obtained, but the image contrast was very low and the noise level rather high, making detailed analysis of the structure difficult. To obtain high-contrast, high-resolution images, hundreds of images had to be collected and carefully processed, one by one. The back projection method was then used to reconstruct a three-dimensional image.
Proteins are extremely susceptible to damage caused by electron beams, so we cannot actually observe the protein as we shoot the image. That means we have to take many shots. We had to get a great many more than 1,000 shots in all, and then make a visual inspection of the resulting images to sort out those that turned out well. However, because of very poor contrast in the images, it takes experience to be able to choose images that can be used for structural analysis. That also took up a lot of our time.
Through this work, not only the structure of the tip portion of the flagellum, but also that of the isolated cap was obtained. The cap was a 5-fold pentamer of hap2 protein and was shaped like a pentagonal table with 5 legs. Flagellum subunits in the filament are arranged in a helical manner, forming indentations at the tip of the flagellum by half-unit staggered binding of neighboring protofilaments. Thus, the binding of the cap and the filament end was achieved over a symmetry mismatch. Along the top layer, made of 11 flagellum subunits along the helical line, there were 4 shallow single indentations and 1 deep double indentation. The cap binds to the filament end by binding 4 of its 5 legs onto the 4 shallow indentations, leaving a large gap open. Despite the solid pentagonal top plate, the 5 legs of the cap appear to change shape in a flexible manner so as to fit into these indentations over the symmetry mismatch. The cap complex can firmly bind to the distal spiral staircase structure of the flagellum, and yet always keep one hollow open to allow a flagellum molecule to polymerize. When a flagellum subunit is bound into position, the cap moves its legs as if it were walking up a spiral staircase. It thus rotates the top plate to prepare the next site for binding flagellum. The cap is thus a rotary assembly nanomachine that effectively assists the self-assembly of flagellum for rapid filament growth. These results were published in Science and featured in the news commentaries of this and other journals.
Flagella are made up of a great number of proteins. Structural analysis of the complex as a whole is important to understand how each of these proteins actually band together, bond, rotate, and grow. Under existing technology, the only way to do so is to use the electron microscope. We want to develop its potential much, much further.
The structural analysis of the motor protein complexes, which are membrane proteins, is also being conducted. How does the motor rotate smoothly by proton flow, even at an energy level comparable to the thermal noise? And why does the bushing work well without lubrication? To find answers to such questions, these membrane proteins are being produced on a massive scale in E. coli, and new purification methods are being developed. Sufficient amounts of these membrane proteins can now be obtained for structural study. Improving electron microscopic analysis resolution is needed for more detailed examination of the structures of protein complexes. An electron cryomicroscope equipped with a cold cathode electrode gun of high brilliance and an energy filter to eliminate inelastically scattered electrons was set up to examine its high-resolution imaging power. The reduction of the noise level in the electron diffraction pattern, for example, was beyond expectation, even at a resolution beyond 3 angstrom. Therefore, high-resolution data can be obtained at a much higher quality, and the three-dimensional structures of various portions of the flagellum are now being studied. As one example, the structure of the rotor has been clarified. To understand the mechanism of the highly efficient proton motor, the rotation speed and its fluctuation or stepping motion must be examined. Many technical issues have to be overcome for this purpose. To measure the motor's efficiency of energy conversion, the energy input and output also have to be accurately measured.
Although it was said that the rotation of the flagellar motor was measured, what had actually been observed was the rotation of the helical filament. But the flagellar filaments are somewhat flexible, so even if we measure their rotation, the data do not represent the actual behavior of the motor itself. In observing the elementary step of rotation, the flexibility of the filament gets in the way.
A new optical system incorporating a specially designed high-sensitivity quadrant photosensor was developed for measuring single flagellar motor rotation. This system is capable of measuring the angular velocity of motor rotation by determining at every 0.2 milliseconds the position of a 40 nanometer fluorescent bead attached to a filamentless flagellar hook. The accuracy of bead positioning is approximately 1 nanometer, allowing the observation of speed fluctuation even within each revolution of the motor. Data indicate that the motor rotation has unexpectedly large fluctuations, stopping occasionally for several milliseconds, restarting within 1 millisecond, and so on. These are new and important clues to understanding the motor's function and mechanism. The mechanism of torque generation is also a major research target. It is essential to clarify the relation between the energy input and output by simultaneous measurement of motor rotation and proton current under given potential differences across the cell membrane. For this purpose, an electrical current measurement system for measuring the proton current has been built. It can measure electrical currents as low as several tens of femtoamperes or 10 to the minus 14th ampere. Dr. Namba believes that advances in nanotechnologies and nanosciences based on the knowledge of bio-nanomachines will eventually greatly contribute to humankind.
Current nanotechnology development is aiming, for example, at a massive integration of data storage devices. Storing all the information in the National Diet Library within a lump of sugar simply by making the size of solid-state devices ultimately small. However, we are trying to reveal something that works under completely different principles. Succeeding in this will enable us to create something that will dramatically change our lives. Conservation of natural resources is one of today's issues, and people speak of the importance of recycling energy. I think that the future of humankind would be much, much brighter if it becomes possible to utilize the extremely efficient energy transduction mechanism used by bio-nanomachines.
Through such efforts as devising new methods of X-ray structural analysis, making further improvements in the resolving power of electron cryomicroscopes, and developing nanophotometry optics for single motor measurement, the Protonic Nanomachine project is making steady progress towards the further understanding of the bio-nanomachine mechanisms.